It's my pleasure today to introduce you to Dr. Tancredi Pentamali. Tancredi actually finished his MD in 2020 from University of Rome, La Spezia, and he did his MD thesis in rare variants in pediatric inflammatory bowel disease. I actually didn't know about pediatric IBD. Then he joined Nicholas Rejewski lab in Berlin in 2018, first as an intern and then as a PhD focusing on single-cell biology. He actually teaches single-cell RNA-seq data analysis to medical biology students and computer science students at the Humboldt University of Berlin. So he's been doing that since 2021. And then you're going to hear about some amazing spatial biology that Tancredi is doing today. And this is for all you spatial biology people out there. Just jump in. Everything's a discovery. Everything's brand new. And you're going to hear from Tancredi this morning. So it's great to have you with us, Tancredi.
Thank you so much for the introduction. Today, I will present you with a three-dimensional approach to spatial biology. We will dive into non-small cell lung cancer and what we can learn from a single patient in 3 dimensions. So most studies actually look at tissues in 2 dimensions, but cells live and interact in 3 dimensions. And these interactions are especially critical in the tumor microenvironment where we have tumor, stromal, and immune cells interacting. And indeed, when we target therapeutically these interactions, this resulted in great advancement in the care of solid tumors with immune checkpoint inhibitors. And this is particularly important because personalized approaches now are critical to identify which patients will benefit from therapy and which instead will not respond or even have toxicities from these very expensive therapeutic approaches. And indeed, in the era of personalized medicine, it's particularly important to understand what will be the efficacy of these approaches. And so we asked ourselves if 3-dimensional single-cell resolved spatial transcriptomics could help us understand what could be drug targets in this patient. So we selected one particularly interesting case of non-small cell lung cancer in which the patient had a very small tumor, was an early stage tumor, but nonetheless, despite chemotherapy and complete resection of the tumor, the patient underwent brain metastasis and very poor prognosis and outcome, only 11 months after the diagnosis. We designed our data collection in this way. We selected 6 sections to be analyzed with cosmics, and we left every time 5 sections intervening between 2 sections that we collected. This gave us the opportunity to increase the Z space that we analyzed, and at the same time, it gave us sections that we collected for additional analysis, such as H&E morphology. We also performed second harmonic imaging to study the composition of the extracellular matrix, and then also multiplex IF and so on. I will not present this data today, and I will focus mainly on the cosmics analysis. So we were left with the problem of where do we focus our region of interest? And to do so, we are collaborating with a digital pathology company called AI Agnostics in Berlin. This allowed us to identify through semantic segmentation where the carcinoma cells were placed and where the tumor-associated stroma was placed. And in this way, we could select a region that was particularly rich for both of them. And then we just sent our sample to NanoString in Seattle, and they were so good in capturing exactly the same region in all the sections so that then we could reconstruct this in 3 dimensions. First thing, we wanted to check. For instance, we have in the same section pan-cytokeratin staining and staining for keratin 19, one of the cytokeratins at the mRNA level. And I think you can appreciate how well the mRNA and protein staining really match. And this is true both for healthy epithelial cells, as we see in the upper part of this field of view, and also for carcinoma cells in the bottom. And you can also appreciate the type of data that we get. So we have stainings for 960 genes, for transcripts of 960 genes, and then we can map them back to single cells thanks to cell segmentation. This basically let us have data that has single cell spatial resolution, and it's very different from the traditional histology. Nonetheless, we can see how well it matches in consecutive sections between the traditional H&E morphology and what we get out of cosmics. We can map more than 100 million transcripts from the 960 genes of interest in more than 300,000 single cells in the 6 sections that we analyzed. We can then use transcriptomic clustering, a typical single cell workflow, to identify cell types. And we identify 18 different cell types, including tumor cells, epithelial cells that are normally present in the lung, such as the respiratory epithelium, the basal cells, the alveolar cells, and then numerous stromal cells, fibroblasts, but also vascular lymphatic endothelium, smooth muscle cells, pericytes, and also diverse immune population, both in the innate and the adaptive compartments, such as macrophages, but also cytotoxic and regulatory T cells, B cells, plasma cells, and so on. We then went on to perform a three-dimensional reconstruction of our data. And to do so, we use STIM, which is a software that we developed in collaboration with Stefan Preibisch, that transforms spatial transcriptomics data, treats spatial transcriptomics data as an image, and therefore allows to apply all the conventional image registration approaches. Here I'm highlighting in blue the respiratory epithelium. So we selected a region that not only showed tumor and stromal cells, but also showed airways, because we reasoned that the presence of airways could allow us to understand if the three-dimensional reconstruction was working, and also will guide the 3D reconstruction in this sample. Indeed, I think you can nicely appreciate how the airways crosses all the consecutive sections along the Z-plane, and then if now we tilt 90 degrees, how we can visualize even the lumen of the airway. We can really look through it in the picture on the right. Then we went on to leverage the three-dimensionalities of this data to define 3D cellular neighborhood. To do so, we picked a radius of 50 micron center-to-center distance. And we went on to identify for each center cell all the cells that were present in the neighborhood. We then quantify the number of cells per cell type in each cell neighborhood, and this led us to the generation of a three-dimensional neighborhood matrix. So basically now we have a new data modality together with the transcriptomic information to, for instance, perform clustering of the cells. We can now group cells based on the similarity of the cellular neighborhood, of the composition of the cellular neighborhood, and we can then annotate these clusters, which we call multicellular tissue niches, and we can identify niches that are rich in tumor cells, such as the tumor core and the tumor surface, and niches that are rich in respiratory epithelium and basal epithelial cells, the airways, in alveolar cells, the alveolar spaces. Then we can distinguish the desmoplastic from the vascular stroma, with, in both cases, having a high content of fibroblasts, but the desmoplastic stroma also showing plasma cells, while the vascular stroma also showing vascular endothelium and pericytes. We can then identify two different immune niches, the macrophage aggregates, the macrophage islands and the T cell aggregates, respectively rich in macrophages and in T cells. And interestingly, in the T cell aggregates, we also find numerous cytotoxic but also regulatory T cells, dendritic cells, and B cells. We can now look at the spatial distribution of the niches, and we can compare this to a typical annotation from an experienced lung pathologist of the H&E image. What we can identify is a nice matching between the two types of annotation. However, the pathologist only annotates tumor cells, while we can actually stratify the tumor bed into tumor core and tumor surface. And also, the pathologist annotates the stroma, which we can further subclassify into desmoplastic and vascular, and identifies immune cells, which we can actually distinguish when they localize in T cell aggregates and in macrophage islands. Then we wanted to really study the spatial arrangement of these niches in 3 dimensions, and I hope you can appreciate how well the tumor surface covers the tumor core, and also the spatial distribution of the different immune niches in 3 dimensions in this 3D rendering of the data. We then focused on tumor cells and identified that actually up to 1/4 of tumor cells were located outside of the tumor bed, meaning the tumor extending beyond the tumor core and the tumor surface. And this is very important because we can now study these infiltrating tumor cells at the molecular level and identify which pathways are upregulated during tumor invasion, which of course is of critical importance for patients because this is what leads to metastatic spreading of the tumor, and then is of course, which is very bad news for the patient. When we compared gene expression in infiltrating tumor cells versus cells in the tumor core, we identify a simultaneous upregulation of vimentin and downregulation of EpCAM and CDH1, E-cadherin, among others. These are key markers for epithelial-to-mesenchymal transition, and therefore we went on to define an EMT score for every cell, for every tumor cell in our data set. And we identify in red tumor cells that have a mesenchymal phenotype, and in blue cells that have a more epithelial phenotype. And of course we see that cells extending outside of the tumor bed have a mesenchymal phenotype, of an invasive phenotype, but we also identify these cells in one specific region of the tumor bed, what we call the EMT focus, that is highlighted here with a box. And again, once we define a region now, we can study what's happening at the molecular level in this region. And we can do this with single cell resolution and also with cell type resolution. So we can look, for instance, at which changes are specific for tumor cells in this region compared to tumor cells outside of the region. And we can do the same for fibroblasts. What we identified was an upregulation of galectin-1, which is specifically expressed in tumor cells in this region. And then we identified at the stromal level an upregulation of IGF-binding protein 5. And this was particularly interesting because at the same time we detect a strong downregulation of IGF-binding protein 7. These are 2 critical factors that regulate the availability of IGF-1. We then went on to visualize these in 3 dimensions, and here you can nicely see what we mean with EMT focus is a specific region of the tumor bed where EMT is and tumor invasion is starting. And we can then show how galectin-1 expression in tumor cells is highly concentrated and specifically expressed only in this region, and this could be a very interesting factor because it has been associated with the remodeling of the microenvironment, the suppression of immune responses. And this is instead the expression of IGF binding protein 5 in 3 dimensions in fibroblasts and of IGF binding protein 7. Interestingly, we detect a strong expression of IGF-1, which is the ligand for these binding proteins, in fibroblasts present in the desmoplastic stroma where the tumor cells infiltrate. So we were able to link the IGF pathway with tumor invasion. Of course, it's very difficult from a molecular characterization to understand the molecular connections, but we see a spatial organization of signaling that is very curious and very interesting for us. Then we went on and asked ourselves, can we use this spatially informed molecular signal as biomarker for patient prognosis? So we went back to bulk RNA-seq in the TCGA, which is a large collection of gene expression and survival data, including lung adenocarcinoma patients, 500 of those. And we actually scored each patient for the enrichment of both galectin-1 and IGF binding protein 5. And in this way, we could distinguish patients with low and high expression of these 2 genes and look at the survival of these genes. And it turns out that indeed, patients that have an enrichment for these two genes have a poorer prognosis with an odds ratio of 1.5, so basically 50% worse prognosis than patients that do not show these molecular features. So we were able to connect what we observed at the molecular and spatial level in one patient with a larger cohort. Then we went on, and to connect with what I was mentioning at the beginning, we went on to study really molecular interaction between cells. To do so, we still leverage three-dimensional neighborhoods defining a 50 micron radius, and we looked for each cell, let's say cell A, to all the pairs of interacting cells in the neighborhood. For each pair, we define a spatial ligand activity score, which is dependent on the co-expression of the ligand and all its receptors in the recipient cell. And then we simply sum the pair scores for each cell, and in this way we can generate a third data modality. So now we have for each cell the gene expression, the neighborhood composition, and the spatial activity score for multiple ligands. We can therefore ask what's specific to the immune niches and also what's shared between the immune niches. And we identified some signals that are specific for one of the two immune niches, for instance, CCL19 in T-cell aggregates or SPP1 in macrophage islands. But then we also identify multiple signals that are shared between the two niches. Most importantly, what was most shared was galectin-9. And galectin-9 is particularly interesting in lung cancer. Indeed, inhibitor like antibodies targeting TIM3, which is the main receptor of galectin-9, are currently under study and in clinical trials. What I'm showing here is how well this spatial activity score localizes to the immune niches and basically covers most of the immune niches that we have in our sample. Therefore, we present a proof of principle. We first generate a three-dimensional and high-resolution map of the tumor microenvironment in which we characterize more than 1 million transcripts across more than 300,000 cells in 18 different cell types. We then leverage the three-dimensionality to identify tissue niches that match with the pathologist annotation, but that are also give us the opportunity to stratify the tumor bed into core and surface. And then also we can perform a molecular analysis of tumor cells that are located in different tissue niches. This led us to the identification of EMT as a key pathway in tumor invasion, which is a well-known fact, and then allowed us to identify an EMT focus where invasion started. In this focus, we can now study the expression patterns in tumor cells and fibroblasts, leading to the identification of galectin-1 and IGF binding protein 5, which are prognostic in a larger cohort of lung cancer patients. We then went on to identify cell interactions in 3 dimensions, and we identify several immunomodulatory ligands to be shared between immune niches. And we also identify galectin-1, which we think could represent a personalized immunotherapy target in this patient. And with this, I would like to thank all the people involved in this project, the Nikolaus and all the Nikolaus Rojewski lab, especially Nikos Karaiskos for the supervision, Daniel Leon for the 3D rendering of the all these beautiful pictures that you've been seeing, and then also our pathologist collaborator in the Frederick Clausen group, of course NanoString, for giving us early access and for generating this very high quality data set. And with this, I would like to just conclude and leave the video running in the background.